Assalamu alaikum. Hello, my dear students. How are you all? Welcome to another biology class. I'm your biology teacher, MD Zahidul Hasan. So today we are going to learn TCA cycle and electron transport systems. Okay, and all these topics belongs to the chapter plant physiology. Um and the go and in my previous lecture, I talked about glycolysis and the conversion of pyruvate to acetyl CoA. So, and now I'm going to talk about the next two steps of cellular respiration. That means the TCA cycle and electron transport system. Okay. So now let's talk about the TCA cycle. TCA cycle. Here the TCA abbreviation is tricarboxylic acid cycle. So this is known as TCA cycle or tricarboxylic acid cycle. It has two more names. One is citric acid cycle and another one is Kreb cycle. Citric acid cycle because the first stable substrate that produced here in this metabolic pathway is the citric acid. That's why this cycle is also known as the citric acid cycle. And another name is the Krebs cycle because Sir Hans Krebs discovered this metabolic cycle in 1937, and for this he was also awarded with Nobel Prize in 1953. So this is also known as the Krebs cycle. Okay, so the citric acid cycle is also called the central metabolic hub of the cell because there are so many substrates that produced here in this cycle can be used in other metabolic pathways. That's why this is called the central metabolic hub of the cell, and it is a final common pathway for the oxidation of fuel molecules such as amino acid, fatty acids and carbohydrates. Okay, and in eukaryotes the reaction of citric acid cycle it takes place inside the mitochondria, that means in the within the mitochondrial matrix. In contrast the glycolysis which take place in the cytosol. Okay, and now see here, this is the overall citric acid cycle. Though it look so complex, but don't worry, I will describe it step by step. And the goal of this TCA cycle is to generate NADH. See here, this NADH and FADH2, and also this ATP or GTP. Okay, and this NADH and FADH2 it will generate ATP in the electron transport system. I will describe the process of the electron transport system in the later stage of this lecture. Okay. So in my previous lecture, I've already discussed about the acetyl CoA production. See here the acetyl CoA. This acetyl CoA through glycolysis, the glucose produce pyruvate and then pyruvate converts into acetyl CoA. So here is that acetyl CoA. So now this acetyl CoA, this is a 2-carbon compound. It combines with the 4-carbon compound oxaloacetate and also it combines with water molecule. Okay, so when acetyl CoA it combines with the oxaloacetate and with a water molecule, uh, um, and this is a condensation reaction, and this condensation reaction is catalyzed by the enzyme citrate synthase. Okay, and all these molecules, that means the combination of acetyl CoA, oxaloacetate and water molecule, it produce here a citrate molecule. Okay, see here the citrate, and enzyme was the citrate synthase. Okay, see, another important thing is the CoA group of this acetyl CoA. This CoA group, coenzyme A group, is replaced by this water molecule. Okay, so the water when combines with the acetyl CoA, it replaced this CoA group, and so now this citrate is then converted into its isomer, which is isocitrate. See, this citrate converts into its isomer isocitrate by two-step process, and the very first dehydration occurs. See here the dehydration, water molecule released from the citrate, and then it converts into an unstable compound which is called cis-aconitate. Okay, and this cis-aconitate when it produce immediately after its production, it rehydrated again, and then it produce the another isomer of this citrate is the isocitrate. So and all this is that means these two steps are catalyzed by the enzyme aconitase. Okay. Mm. Then the isocitrate dehydrogenated. This isocitrate is dehydrogenated by losing proton, and this proton is picked up by NAD plus, and then the NAD plus turns into NADH. Okay. So, and this thing is catalyzed by the enzyme isocitrate dehydrogenase, and simultaneously another thing happens here, and this is the decarboxylation. Decarboxylation means D means remove, carboxy means carbon dioxide. So decarboxylation means the removal of carbon dioxide. So simultaneously dehydrogenation of the isocitrate and also the decarboxylation of the isocitrate occurs simultaneously. As a result, the isocitrate turns into alpha ketoglutarate. Okay. So, alpha ketoglutarate, this molecule also undergoes actually simultaneously decarboxylation and uh, dehydrogenation. Okay, and this reaction is catalyzed by the enzyme alpha ketoglutarate dehydrogenase complex. Okay. See, at the alpha ketoglutarate molecule is dehydrogenated, and the hydrogen molecule the dehydrogenated, that means it released proton, and those proton is picked up by NAD plus, and it then turns into this NADH, and also the carbon dioxide molecule of carbon dioxide is also released or replaced by the CoA group, coenzyme A group. Okay. And as a result, after this oxidation, dehydrogenation and decarboxylation, this alpha ketoglutarate, it produces the succinyl CoA. Okay. See here the succinyl CoA. So then in the presence of the enzyme succinyl CoA synthetase, the succinyl CoA turns into succinate, okay, by substrate level phosphorylation, and it release the CoA group. Okay, it release the CoA group, succinyl CoA, it release its CoA group, and this thing is actually catalyzed by the enzyme succinyl CoA synthetase. As a result, succinyl CoA turns into succinate. But see, here the substrate level phosphorylation take place. That means the energy that liberated during this process is used in the synthesis of ATP. Okay, in plants ATP, and in case of animal it is GTP. Okay. So then this succinate is dehydrogenated into fumarate. Okay. Dehydrogenation occurs in the presence of the enzyme succinate dehydrogenase, and then this dehydrogenation, that means the release of the released proton is picked up by FAD plus, and then it turns into FADH2. So rather than NAD plus, here the proton is picked up by FAD plus. Okay, so in this step FADH2 molecule is produced. Then this fumarate, after hydration, that when it combines with a water molecule, it then turns into malate, and this reaction is catalyzed by the enzyme fumarase. Okay, so then finally after dehydrogenation by malate dehydrogenase enzyme, this malate is finally converted into oxaloacetate. Okay, and here the hydrogen the proton that actually released from the malate is picked by NAD plus, and then this NAD plus turns into NADH. Okay, so And this oxaloacetate, this is the very fast product of this citric cycle. So the oxaloacetate is then becomes available to combine with the another acetyl CoA to start a new cycle all over again. So the final result of the citric acid cycle is 3 NADH2, one from here, another one from here, another one from here, so 3 NADH2 and FADH2 from here, and a ATP or GTP. So the cycle and this cycle runs two times because we have two molecules of acetyl CoA from 2 pyruvate. So finally, from this citric acid cycle, uh, mm, uh, we have received total 6 NADH2, 2 FADH2, and 2 ATP or JTP from Krebs cycle. Okay. So now the electron transport chain or electron transport system, this is the final step of the aerobic respiration, okay, aerobic cellular respiration, and the electron transport chain is present in the inner membrane of the mitochondria. It consists of various enzymes, coenzymes, cofactors, and electron carrier proteins. The electron transport chain is actually the transfer of electrons from NADH and FADH2 into oxygen via multiple carriers. Okay. So the electrons derived from NADH and FADH2, when it combined with the oxygen, the energy released from the oxidation and reduction reaction is used to derive the synthesis of ATP from ADP. Okay, which is our final goal. That means the ATP production. ATP is the biological currency of a cell. So this transfer of electron is done by multiple carriers which constitute the electron transport chain. Okay, so let's see. Now you see here the you see here that this is the inner membrane uh, inner mitochondrial membrane, and here this bottom region, this portion is the inner mitochondrial matrix, and here this upper portion, this is the intermembrane space. That means the space between the outer membrane of the mitochondria and this inner membrane of the mitochondria. Mitochondria has two membrane, the outer membrane and inner membrane. This one is the inner membrane, this one is the inner membrane, and there is an outer membrane. So this is the space between that outer membrane and this inner membrane, and this is the inside of the mitochondria. That means the matrix of the mitochondria. Okay, so here in the inner membrane, we have a series of enzyme complexes, complex 1, 2, 3, 4, and 5. So, and we have also other two molecules that is not as complex as these complexes. This is first one is the coenzyme Q, and another is the cytochrome C. Okay, so all these things are embedded in the inner membrane. Okay, and here the complex one, and see here the complex one is called the NADH dehydrogenase. Here the dehydrogenation of NADH occurs, take place. It contains this complex one, it also contains FMN means the flavin mononucleotide, and it also it has iron atoms. Okay, and now see here the complex 2 is also called succinate dehydrogenase. It contains FAD and also the iron atoms. And the coenzyme Q, it transfer electron from this complex 1 and from this complex 2 to this complex 3. This is a transporter. It transport the electrons, okay, from this complex 1, from this complex 2 to this complex 3. See here the complex 3, this complex contains cytochrome B and iron atoms and cytochrome C1, and here the cytochrome C, this cytochrome C, it transfer electrons from complex 3 to complex 4. So this one is the another transporter, and here the complex 4, it contains cytochrome A, cytochrome A3. Okay, and lastly the complex 5, this is the ATP synthase. That means this complex 5, actually this ATP synthase, this is an enzyme that synthesize ATP from this ADP plus an inorganic phosphate. Okay, so see, this is the electron transport chain, electron and proton flow of the electron transport chain. Mm, so when the NADH approaches to all this NADH, NADH, FADH, all these things actually it produced from the previous pathways like citric acid cycle, glycolysis, and all other metabolic pathways. All those FADH2, NADH2, they come here, and then NADH, NADH molecule or NADH2 molecule, they approaches to complex 1, and NADH can give up its electron and give up its proton. Okay, and so see here the green line is the proton track, and here the orange line, it indicates the flow of electrons. Okay, so here the NADH, when it give up its electron, here it give up its electron to the complex 1, and it also give up its proton to the complex 1. Okay, in this process it donates electron to complex 1. So in this process, this NADH, it donates its electron to complex 1, and when electron enter into the complex 1, complex 1 gets this complex 1, it gets supercharged. Okay, and when this complex 1 gets supercharged, it has the energy to pump this proton from the mitochondrial matrix into the intermembranous space. Okay, so when NADH gives its electron to the complex 1, then this complex 1 it gets supercharged, and the supercharged complex 1 it gains the energy to pump the proton from the mitochondrial matrix to the intermembrane space. Okay, so as a result, the intermembrane space have much more protons than the inner matrix. Okay, okay, then the C1, then the complex one then pass its electron. See, it pass its electron then to the coenzyme Q, and then coenzyme Q sit there waiting for the further instruction. And now at this point, the FADH2 molecule it comes along and approaches to this smaller complex 2, and then just like NADH, FADH2 then can give up its electron and turns into FAD. Okay, so it donates its electron to the complex 2, and then it donates, so FADH2 it donates its electron to the complex 2, but the complex 2 cannot be supercharged like complex 1 and cannot pump protons from the matrix to intermembrane space. Okay, so this complex 2 then just pass it electron to the this coenzyme Q. So this coenzyme Q it has electrons from complex 1 and also from the complex 2, and this coenzyme Q then pass these electrons to complex 3. Okay, when this complex 3 this receives its receive electrons from coenzyme Q, it gets supercharged. Okay, and then the complex 3 creates enough energy potential to pump the proton from the intermembranal space, sorry, from the matrix to intermembrane space. Okay, so look at here, the intermembrane space getting the accumulation of proton. So there's a much intermembrane accumulation of protons at the accumulation of proton at the intermembrane space than the matrix. Okay, so at this point, this complex 3 pass its electron to cytochrome C, the transporter. Cytochrome C then pass these electrons to the complex 4. Okay, then same thing happens here. The complex 4 it supercharged, and after receiving the electron, then it also pumps proton from the matrix to the intermembrane space. Okay, so here in the intermembrane space, tons of positive charge, that means tons of protons yeah, protons here, see. So this complex 4 then pass this electron to the the electrons they have from the cytochrome C to the final electron um, receiving final electron acceptor, okay, and this is oxygen, okay. This oxygen is the final electron acceptor. It receives the electron from the complex 4 and it receives electron, and also it combines with the protons, and then this oxygen after the when it combines with proton and electron, it turns into water molecule, okay. Then here the ATP synthase comes into play. This ATP synthase make use this proton gradient. See here the lots of protons here in the intermembrane space. Then this matrix region. That's why a proton gradient generates here, and this ATP synthase make use this proton gradient to generate massive amounts of ATP. Okay. So ATP molecule generally, this proton molecules generally moves from the higher energy to lower energy gradient. See, then from this the higher energy gradient and the lower energy gradient. That's why these proton molecules they moves from this intermembrane space to the matrix through this ATP synthase. Okay, so to achieve equilibrium, this proton will flow from the intermembrane space to matrix. Yeah, and then the ATP synthase, when they do this, it is an energy input that catalyzes the conversion of ADP to ATP. ADP and inorganic phosphates they combined by using this proton transfer energy, and then they produce ATP molecules. Okay, thus ATP is produced. So this was our target, taina? So now this is the overview of cellular respiration here. Um, each 2.5 moles of ATP is produced from 1 mole of NADH, and 1.5 moles of ATP is produced from 1 mole of FADH2. As a result, all the ATPs, all the NADH2, FADH2, they produce electron through this electron transport system, which is produced by the glycolysis and the citric acid cycles, and finally the total energy yield is 36 ATP from a single glucose molecule. So in our cell, through cellular respiration, a single glucose molecule produced 36 ATPs. Okay. Now let's see a sample CQ. Yeah, this is a stem in the overall cellular respiration pathways, and see here there are 4 questions, and the mark of these questions are 1, 2, 3 and 4 respectively. You have to define what is ETS, what is electron transport system. Okay, and then you have to write down the names of the enzyme complexes in ETS. Enzyme complexes, that means the complex 1 to complex 5. Okay, you have to write the names only. And then you have to describe the process of ATP production from NADH2 and FADH2. So what is the process of the ATP production from NADH2 and FADH2? Which one? This glycolysis or Kreb cycle or electron transport system? This one. The electron transport system is the process of ATP production from NADH2 and FADH2. And finally, you have to write that you have to describe how kinetic energy is produced in Krebs cycle, starting from pyruvic acid. That means the pyruvic acid, when it enters into the Krebs cycle by producing acetyl CoA, and then it produce NADH2, FADH2. So that means the kinetic energy. So you have to describe the kinetic energy production in this Krebs cycle. Okay, I think that's all for today. Thank you all.